Hi. Yeah, hello. Yes, thanks for the introduction, and thanks for the organizers for letting me present my some of my results here today. So I'm going to talk about the effects of vitamin D supplementation in cellular immunity and mycobacterial killing in cows. And just a brief of introduction. The relationship between vitamin D and tuberculosis has been known for many years. And during the pre-antibiotic era, vitamin D was used as a treatment for tuberculosis in different forms, like in the cold liver oil, after sunlight exposition, or in the phototherapy. However, the discovery of streptomycin and other antibiotics banished the interest in vitamin D. But in the recent years, there has been numerous studies to try to see if there was or not a relationship between vitamin D and tuberculosis. And one of the most comprehensive meta-analysis done in this relationship with different aspects of tuberculosis and vitamin D was done in 2017. It showed that actually vitamin D deficiency is associated with a higher risk to develop tuberculosis. But of course, controversy persists, and there has been numerous studies recently. Most of these studies has been done in vitro, in macrophages, because macrophages is the first line of response against mycobacteria. So mycobacteria is an airborne pathogen that after inhalation reach the lung, and in the lung it encounters the macrophage, alveolar macrophage that will ingest and try to kill and destroy the bacteria. And the research has shown that when you stimulate the macrophage with the active form of vitamin D, 125, infected with mycobacterial tuberculosis, there is a reduction in the number of bacilli in comparison with the cells that were not stimulated. And as Professor Corwin Nelson mentioned, now we know, or we have a good understanding on the molecular mechanism of vitamin D against mycobacteria and other infections. So I'm not gonna talk a lot about that, but we know that after stimulation, macrophage in line with the BDR receptor, there is an induction of different expression of antimicrobial peptides and defensins, and the induction of other mechanisms as autophagy that altogether will help to kill the mycobacteria. However, the response to mycobacteria is very complex. There are different cells that can be infected by bacteria, like macrophages or neutrophils, dendritic cells, for example. And of course, there are different cells from the innate and adaptive immunity that are required to control the infection. And of course, now we know by different research data that vitamin D can modulate some of these cells, and the effects of vitamin D will, of course, the modulation of vitamin D in these cells will affect the outcome to infection. So we wanted to take a more comprehensive approach. So using BCG as a model, we infect peripheral blood leukocytes from cattle, or named PBL. In the PBL, we can find different cell types like macrophages, neutrophils, T cells, different subsets of T cells like CD4 or CD8 cells. Therefore, there is a very complex interaction between cells. So we stimulate them with vitamin D and infected with BCG. And after 24 hours, we observe that the cells that were stimulated had higher bacterial killing in comparison with cells that were not stimulated. Then we wanted to see what was the contribution of each of these cells. So we did a specific depletion of these subsets. So we have here the complex peripheral blood leukocytes, and then we did a depletion of macrophages, neutrophils, T cells, CD4 and CD8 cells. And we observed that After stimulation with vitamin D, neither the depletion of macrophages nor granulocytes affected the effect of vitamin D. Therefore, this means that vitamin D is modulating both cells, and the bactericidal effect is of the effect of both cells of vitamin D is required to control infection. So as you can see, the mycobacterial response, or the immune response to mycobacteria, is very complex. And the only way to really assess all these interactions is by in vivo studies. So we did a long-term in vivo supplementation in cows from birth to 7 months with 3 aims. The first one was to develop a model to drive divergent vitamin D status, then to assess how this variation in the vitamin D levels affected the cellular immunity, and later on to see how these affect the mycobacterial killing ability of the cells. Just briefly, we gathered 48 cows, and we allocated in 4 groups: control and vitamin D in, and control and vitamin D out. The control groups were supplemented with the standard dose of vitamin D, and the vitamin D groups were supplemented with the highest concentration of vitamin D within the current European guidelines. And then the in groups were kept indoors all the time, and the out groups were moved outdoors after weaning. Here in the graph below, we have the vitamin D profile of all the cows through the 7 months. The red line is showing the optimal threshold of vitamin D for cows, that is 13 nanomoles per ml. And the arrow is showing the moment where the animals were moved outdoors. So here we observe a high incidence of vitamin D deficiency at birth that was not overcome in the pre-weaning period. But later on, after post-weaning, we observe a higher divergence on the vitamin D status within the cows, with higher levels of vitamin D in the outdoor groups, and as well a high level of vitamin D in the vitamin D indoor groups. In contrast, the control indoor groups has lower vitamin D levels through all the study. Then we assess the changes in the cellular immune profile by counting the cell counts of neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And we did a multifactorial factor analysis. So here you can see that the groups, the outdoor groups, are very similar because they are close together between them, whereas the indoor groups are very different because the distance between them is higher, meaning that the overall cellular immune profile is different over the time. And the control indoor groups is separated away the dimension 2. Dimension 2 is described by the difference in the neutrophils and in the eosinophils. This is because the higher distance of the cells. Furthermore, the postdoctoral research from our group, Dr. Sayar Bab, has also looked at the differences of the vitamin D effects on other cell types. His focus on the analysis of the gamma delta T cells. Gamma delta T cells are very important for the early innate immune response in cows. And he has shown that vitamin D also affects the proportion of gamma delta T cells in animals with divergent vitamin D status. Finally, we assess the changes in the mycobacterial. For this, we assess, we took the animals from the indoor groups that they have different vitamin D levels, with higher levels in the vitamin D indoor groups. And with our same model of peripheral blood leukocytes, we infected them with BCG in vitro, and we observed a higher BCG killing after 24 hours of incubation. And this was associated with a lower inflammatory response measured as the production of IgG1. Of course, we need a more comprehensive overview of this response. So we are going to do a high-scale gene expression profiling from these animals to have identification of different biomarkers. So just to conclude, I have shown you that we have developed a model to try the divergent vitamin D status within the current European guidelines. This model allows us to analyze the effects of vitamin D deficiency in calves, in cellular immunity, in mycobacterial killing, and in microbiome. That is the next talk with Stafford. And I hope I have shown you that vitamin D model is an attractive alternative to study the effects of vitamin D in a One Health approach. Finally, I just want to thank my supervisors, Professor Kian Mead, Steve Gordon, as well as the team members, my sponsorships in Mexico, NIFAP and CONACYT. And I would like to give a special thanks to all the people in Chagas that helped us to carry out this in vivo trial during the pandemic. And of course, thanks to all of you for your attention.